Thank you very much. This is a wonderful event. My name is Jack Elias. I'm the Dean of Medicine and Biology. I want to wish everybody a good morning and thank you all for coming today. It's really a pleasure to welcome you to this Day of Biology. This is part of our celebration of Brown's 250th anniversary. For those of you who weren't here last year, that's what it looked like up on the green as we kicked off the 250th. You'll notice a couple things. First, there's active fireworks over wooden buildings, and so there we have a fire alarm message. You will also notice that there was much less snow last year than this year for reasons that only a meteorologist can explain to you. But seriously, today you're going to have an opportunity to hear from some of our most distinguished alumni and faculty during the lectures and panels. And it's a very exciting event, and it's a very wonderful event. I wish you could all be where I am and look out over this audience of people who are all here for the same exciting reason. On the other hand, it's a bittersweet event. The Day of Biology is dedicated in the memory of Associate Dean Marjorie Thompson. Dean T was actively involved in actually planning these events when she passed away last September, and we'll talk more about her in this afternoon events, but we are deeply saddened that she is not here today. The Walter J. Kenworthy PhD Keynote Address will start things off for us. Let me tell you a little bit about the man that the lecture is named for. Dr. Kenworthy earned his bachelor's degree at Temple University and served in the Army during World War II. After the war, he completed his PhD at the University of Pennsylvania, and in 1952, he accepted a position at Brown and moved with his wife and 2 sons to Providence. He taught at Brown for 12 years until he became dean at the College of Wheaton College in Norton, Massachusetts. And then in 1972, he became the vice president of the Exxon Education Foundation in Pelham, New York. When he passed away in April last year, he was remembered as an intellectual who dedicated his life to learning, to his family and friends, and to his country. Dr. Kenworthy's sons are here with us today, James and his wife, Catherine, as well as their 2 sons, Charles and Stephen, and his other son, Paul, with his wife, Laura. Can you just wave or say hello? Here we are. Thank you. Thank you for joining us today. Dr. Kenworthy is also remembered as a dedicated teacher and mentor. One of his former students, Dr. Gordon Cohen from the Brown class of 1959, remained close to him for over 50 years. It's because of the generous gift from Dr. Cohen and the memory of Dr. Kenworthy that we're able to bring this lecture and the Day of Biology to you. One of the things we often talk about here at Brown is the dedication of our faculty to the teaching of students. Here is an example of an interaction that's lasted for 50 years, a truly Brown-like event. This friendship, which lasted 50 years, is really worth celebrating. We're fortunate to have Dr. Cohen and his family with us. Wave, they're sitting right here in the front. Thank you very much. Today's event would not be possible without the over 30 panelists and speakers who are here today, one of which I just talked to in the hall, flew in from sunny San Diego to join us and is shivering somewhere in the audience right now. Now it's my pleasure to thank all of these people, but it's also my pleasure to introduce our first speaker, which is the Walter J. Kenworthy lecturer, who is Dr. Craig Mello. When you hear about Dr. Mello, you're not surprised that people at Brown call him one of our own. He was a biochemistry concentrator. He received his bachelor's degree from Brown in 1982. He completed his PhD at Harvard in 1990, and 8 years later, he and his colleagues published the definitive paper in Nature that would change science, change therapeutics, and change Dr. Mello's life. He and Andrew Fire Stanford discovered the phenomena of RNA interference. This work has really modified our understanding of what RNA actually does and the role that it plays, not just as a messenger, but in the expression of genetic information. This discovery allows scientists to turn genes on and off. It's become a tool that's essential to understand what genes do and how they do it, and it's a technique that's evolved into drug development and influenced all sorts of diseases. The journal Science called their discovery the breakthrough of the year in 2002, and in 2006, Dr. Mello and Dr. Fire shared the Nobel Prize in Physiology or Medicine for their discovery. Dr. Mello is a Howard Hughes investigator at the University of Massachusetts Medical School, where he has been since 1995, and he is co-director of their RNA Therapeutics Institute. The title of his talk today that I have is RNA Memories, Secrets of Inheritance and Immortality. Please welcome Dr. Mello.
Hello, everyone. It's great to be here. It's great to be back. I don't know if I had any classes in here, but I probably at that time never thought I'd be lecturing in one of them. But it's really great to be here. And I want to just say that I was really honored to be giving the Kenworthy lecture because my dad took his only biology course at Brown from Dr. Kenworthy. I have a lot to owe to Brown, actually, I have to say, because my mom and dad met here, and I wouldn't be here if it wasn't for that. He took biology. He was a geology concentrator from Dr. Kenworthy. I think it was a zoology course, and he became so fascinated by biology that he became a paleontologist, and then he took a job at the Smithsonian after he got his PhD, where I got totally blown away by the history of the Earth and of life, and just wanted to be a scientist since as young as I can remember. So anyway, it's great to be here. I'm going to tell you about our research and about I think most of my talk is really meant to give you an overview of how exciting biology is right now. But I think there's a really fascinating field in biology that has to do with the flow of information from one organism to another, and also some of the I guess you could call it immunology that organisms have to have. That's self-defense against movement of that information. But there's also useful information moving. It's really a fascinating area right now in biology. And I'm going to just show you this little movie because it illustrates something. These are microbes in this the little colors in this man here. And it turns out that our genome is only about 1/100th of the genetic information that's in and around our bodies. I don't know, maybe that didn't play right. But anyway, there's a little animation that you can get from on NPR about how in order to be healthy, we need to have these microbes. You can't live without them. We're actually not really even, this is true for many organisms, we're not really just a single species. We are commensalists, and we need to have these other species in order for us to be healthy. So you're not alone, and in fact, you're not even really you, if you think about it. There's some other, I think my slide projectors, my movies are not playing right, and now my slides are not behaving. All right, let's see if I can go back. All right, come on. Here it goes. So there's a lot of information flowing, we call it horizontally. You've heard about your genome, you inherit that from your parents, but you also get information from viruses, and organisms like bacteria can also transfer information into other cells, including plants and animals. Can we have the lights down a little bit? This slide here is showing viruses in seawater, and the big green dots are algae, and the tiny little green dots are viruses called bacteriophage that are seen in this seawater. And there's estimated to be about an average of 10 to the 7 viral particles per milliliter of water on the planet, which is a huge number. It's like 10 to the 32, and that's just a tremendous amount. So the viruses, they're the most abundant life form on the planet. And the fact is that none of the animals or plants would be here if it wasn't for the fact that the viruses need us, so you should be happy. Again, you know, it's not simply that they're pathogenic either. They're actually doing some really important things probably to help the flow of information and to transfer information from organism to organism. The EMs on the right show you what these things look like. They're incredibly tiny, of course, but they have this, these amazing particle structures to them. And you're looking at right here one of the most abundant forms of, you can't almost call it life, but you know, biological activity on the planet. It's really, really fascinating. And I'm going to get to, in a moment, oh, here's another picture. This is an artist's picture, what these might look like actually down at the, almost at the atomic level here. You're looking at these structures, and they're landing on a cell. They're a bacterial cell and injecting their genetic information into it. You inadvertently drink millions of these a day. You're exposed to them constantly. They're really fascinating. Of course, there are other kinds of viruses that actively infect people, and usually you think of them as bad, but they're actually now, you know, there are many examples of genes that are transferred by viruses. In fact, your genome is about 8% viral DNA, and it's about 50% of what are called transposons, which are viruses which have lost their ability to move from organism to organism, and they're trapped in the human germline. So your DNA is actually more than 50%, you know, remnants of genetically mobile elements. Oh, and you can buy that. Since I used his picture, I thought it should put it up there. So germlines, and I'm going to tell you what a germline is in a moment. This is a movie in the background of one, exist on a cosmic timescale. So every animal and plant on this planet is related to each other. We're all related to each other, of course, as humans, but we're also related to grass and E. coli. And you're looking at a germline here where these are what we call the nuclei of the this is an ovary, and these are the forming eggs, and this is a C. elegans, a nematode ovary. Our germline and the germline of nematodes journeyed together on this planet for 3 billion years. Okay, so the history, the common history of the biology and the biological mechanisms that make up the ability to make an embryo, an egg, is incredibly ancient. So we can learn a lot about our own biology by studying the biology of other animals. So that's one of the reasons people study this worm, which I'll tell you more about in a moment. So here's the cell, and the green stuff that you're seeing here is mitochondria. And mitochondria are the remnants of a bacterial infection that occurred over 1 billion years ago. And instead of being destroyed like most infections are when they get into an organism, they took up shop, and now they provide an essential function for animals. So animals and bacteria are now living together and helping each other. And in fact, these guys, these green things, have given a lot of their DNA to our nucleus, you know, if you will, into the center, into the middle. This big black thing here has all the other genes in it. A lot of the genes from these bacteria are now in this nucleus, where they're copied and replicated that way. And they're also replicated and inherited here, and you can see how there's a flow of these into the eggs. So the germline consists of the genomic information in the nucleus, and then a lot of information in the cytoplasm that you can see flowing in the form of the mitochondria, There's other things in here. Look at these red things. Do you see these red dots? These are germline nuage, we call them, and inside these, they're RNA, and lots of other interesting things that I'll be telling you about in a moment. A lot of jargon that you don't know what I'm saying when I say it, so I'm going to get to that in a minute, I hope. So RNA, let's start with the first bit of jargon. I was giving a lecture once, and I think I was talking to a reporter, and the reporter said, well, you know, my listeners don't know what RNA is. Can I call it protein? And I said, no, sorry, you can't. So this is just to give you a little, to play up the importance of RNA. I just stole this from
Every creature,
Nova
you know this
Science
from
Now.
school, is made from a recipe that comes from its DNA, spelled out in chemicals, A's and C's and T's and G's, inside the famous double helix. Every creature has its own DNA, different from mice, and then for whales, and for flowers, but to go from a chemical recipe, A's, G's, and T's, to a real creature that squeaks, or soars through the air, or turns gloriously pink, that requires RNA. RNA is the thing that turns you from a chemical code to a real, pulsing, living creature. RNA builds life. That's big.
Okay, so a lot of information was just up there for a minute, and it's actually really essential that you guys all get this. So I'm going to have one more sort of background movie that I'll get to in a moment. But there's DNA, there's RNA, and there's protein. Gene expression is really simple. It's like maybe the simplest part of biology is gene expression, because we only have 4 letters in the alphabet. And all organisms, including those viruses I showed you, including, you know, the plant and the bear and the birds in that movie, we all have the same 4 letters in the alphabet of our genetic code. And the beauty of that double helical DNA structure is that the G in the nucleotide in the DNA called G and C, they always pair. A and T, they always pair. And you have this beautiful ladder staircase structure where these 2 molecules pair with each other in this chemically really precise way. And then when they separate, they can serve as templates to copy the other strand. This was what was so amazing about the discovery of the double helical structure of DNA, because immediately it gave us an explanation for how you could replicate the genetic information. Okay, so you have replication of the DNA, and then the DNA can express the genes by making an RNA copy. So it's, again, just 4 letters give you the RNA copy of the DNA, and it's just organized very slightly differently, but we have to call it something different because it is. It's called RNA. The RNA is made, and then the RNA gets translated in the cytoplasm of the cell by something called the ribosome. The ribosome is able to take the genetic information and read it 3 nucleotides at a time and incorporate amino acids into a chain using that information. I'm going to go over this again, but you really have to know this in order to understand anything I say. So I've decided I have to do this. We're going to fly inside of a cell. Now that's the nucleus. We're going in there. And now we're flying along. The DNA is all coiled up inside the cell, and when a gene needs to be expressed, there's a polymerase that can get on there. The DNA can open up. Now I stopped it right there because if that polymerase was going to make a copy of the DNA, it would be a DNA polymerase. It would read the genetic information there, and it would make a new strand that's identical to that strand. And then another one would copy this strand, and now you'd have 2 copies of the DNA. You'd have 2 copies of what we call the genome. But here, this green blob is, in this movie, is RNA polymerase. So it's going to read the genetic information here and make an RNA, okay? And you can see that there are different colors here. The purple and the orange pair up, and the blue, and I guess that's yellow, pair up. That's why the information in this strand specifies the information in that strand. Okay, and then this guy, I'll let the movie keep playing, and you can see what happens. It's going to make an RNA copy The nucleotides don't actually look like this. And there's a few things wrong with this movie, but it's okay. So this is the mRNA getting made, and the mRNA gets processed and then it gets transported out into the cytoplasm where it's going to be translated, and you'll see that happen in a moment. And RNA interference is a process that I'll get into in a moment where we can interfere with the expression of this mRNA. Once it's out there in the cytoplasm, here's the ribosome. It's reading the information here and incorporating these amino acids. And I'll get to this in a moment too. Amino acids are very complicated. That's a scientist injecting double-stranded RNA, and this is the beginning of the RNA interference mechanism. We call this an siRNA, or a small interfering RNA, and this is an enzyme that uses the genetic information in this piece of RNA, this short piece, to find matching information, and then it can cut it. It's really very cool what this stuff can do. Now, one of the big things that's wrong with this movie is that everything is slowed down tremendously. So the polymerization that you saw, the green blob on that first part involved in transcribing the genetic information in the DNA can move, it can incorporate about 50 nucleotides per second into that chain, and it does it flawlessly. There are some DNA polymerases that can go 1,000 nucleotides per second. So cells are incredibly sophisticated in their ability to handle information. The translation machinery can translate that information into an amino acid chain at a rate of about 20 amino acids per second. And again, it's done almost flawlessly. So the really interesting thing here, again, about gene expression, really simple. 4 letters in the alphabet. There's only 20 different words. The words are the amino acids. Amino acids are all different. They have different chemistry. They're specified by 3 nucleotides of genetic information, which are read one at a time by the ribosome. Ribosome will go along 3 nucleotides at a time, find the appropriate amino acid whose, I can't get into all this, but it's a really cool stuff that's going on in there. Is like these things called transfer RNAs are involved in bringing the amino acids in one at a time into the ribosome. It's fascinating, but it moves along flawlessly incorporating those amino acids into that chain. The proteins then carry out functions for your cell. RNAs also do that, but proteins are kind of like the workhorses that carry out different functions. Proteins are capable of incredibly different chemistries. Nucleic acids, as you can see, are really simple. They tend to be, in these relatively simple structures. Don't get me wrong, they can do lots of folding and other things, but proteins are really diverse. And even supercomputers can't predict how a particular stretch of amino acids are going to fold up and form a protein. We have to crystallize the protein and study them with all kinds of high-powered techniques to really figure out what a protein looks like. But anyway, that's sort of a primer on gene expression. It is actually really, really cool, and everybody needs to know this. It's like if you're against genetically modified food, you have to know what genetics is or what genes are and DNA and how it works. Whatever, everyone's got to know this. You've got to know this stuff. And it's the same in them, in the worms, as it is in us. It's exactly the same. It's like almost entirely, all the basics of gene expression were worked out billions of years ago, so all the organisms alive use the same rules. This is going to be a really long lecture. I can see that. I think I'm on slide number 4. This is the real prize winner. This is called C. elegans, and they're a great system. They're only about the size of a comma on a printed page. Here's the ovary that I showed you earlier. They're really amazing organisms because they start out as a male. They make sperm. And then they stop making sperm, and they save those in the spermatheca, and they switch to being female, and then they make eggs, and then they fertilize those eggs so that one animal, if you put it on a petri dish, can make 300 progeny in 3 days. And if they could eat us, we wouldn't be here. So And actually, some of them do parasitize humans, so they're actually a big problem. Not C. elegans, but other types of nematodes are a big problem in areas where there's not clean drinking water. But we can learn an awful lot about ourselves from this organism because it has a brain up here, it has 300 neurons, it has an intestine, skin, of course, germline, much like our own. And we can learn an awful lot. That's why this animal has won 6 Nobel Prizes. You know, it's probably going to win some more too, because it's really a powerful system. Now, I told you that protein, when it's made, after that ribosome, you know, incorporates these chemical building blocks, the protein folds up, and it can do all kinds of interesting folding. This is a protein here shown folded up, and you can see that, you know, this part of the protein has formed like an alpha helix, it's called, and these are beta sheets. They're like just, you know, folding of these things into this really elaborate, interesting structure. And this is a protein we call an argonaute, and in the middle of this argonaute, you can see the basically an RNA here in red. That's the guide RNA, and the blue, which is the target RNA. If you remember in the movie, that thing that was searching with a little search query in it, that's this protein. And this is what we discovered when we were working on RNA interference. We discovered Google. Cells need a search engine because they're dealing with information in much the same way that we all deal with information on the World Wide Web. And what you see here is a search query in red, and the target of that search query, which would be a longer RNA, it would go out of the field of view here, actually. You can see this precise pairing that goes on between the query and the target. That precise chemical information allows the query to precisely identify a matching RNA in the cell, and only the one that matches up perfectly will be able to undergo those elaborate base pairing interactions, just like the base pairing interactions that allow the 2 DNA strands to form that spiral staircase of the DNA. RNA can do that too. And this protein uses that kind of pairing information to find matching information in the cell, and it only needs a short sequence to find the perfect match, right? Just like when you search the web, you don't need to know the whole thing you're looking for. You just need to know a couple of key words, and you can find everything you need. It turns out just 20 nucleotides of information is enough to uniquely identify almost any sequence, almost any gene in the human. And so these proteins are search engines. And it's what was really exciting back in 1998 when Andy and I published our paper describing the mechanism that involves these proteins, is that we learned how to enter search queries into the search engines inside of cells. And humans, we've discovered this protein in the worm. Humans have it, plants have it, fungi have it. There are even bacteria that have this same type of search engine. And it's really turned out to be a powerful tool, because now we can use the search machinery inside of cells to find genes and to regulate them. And it's really transforming what we can do in the laboratory. Oh, and here, and this enzyme has a catalytic center where the target strand can then be cleaved, and then the search query strand is retained, and this protein can go out and find another target. Oh, yeah. The other thing about this is there are versions of the search engine that allow misspellings. Don't you love it? If you had to be a perfect speller, I don't think I could use Google. Half the time I don't know how to spell the word, and I just type in whatever seems close, and I still get what I want. Cells do the same thing. In fact, mismatched pairing in that search engine is really powerful because it allows a mismatched query to regulate multiple different targets that are similar but not identical. Now here's another organism. We discovered this very fundamental mechanism, of using search engines, that cells do this. We learned this first in work in C. elegans, but there was also beautiful work on related mechanisms in fungi and plants. So model organisms can be really powerful in terms of breaking open and understanding concepts in gene expression. Here's another organism that is a bacterial cell where the following system was discovered. And you may recognize this from earlier in my talk. This is a virus infecting a bacterial cell, injecting its DNA, and then this virus will try to take over the cellular machinery to make more viruses, and if it succeeds, then the cell's in trouble. But what the cell has done is it's developed a mechanism for taking the viral information, chopping it into pieces, and inserting pieces of the virus into this, what's called a Crispr array. So if this cell is able to survive this viral infection, it will now have a little chunk of the viral DNA inserted at this locus. And then it can re-express this piece of viral DNA to make little guide RNAs that get loaded onto a search engine that's very similar but actually independently evolved from the Argonaut system. And this will then search for matching information, this time in the DNA, and if it finds a good match, it'll cleave it. So this is what this protein looks like. Again, I told you proteins fold up into elaborate structures, and you're just seeing a movie here based on the structural biology of this enzyme now called Cas9. This is from Jennifer Doudna's lab. You can see the RNA guide in the middle of this, and this protein will find information in the DNA of it'll find target information that matches the RNA strand here, the guide RNA, I think shown here in red, and it'll open up the DNA, allow the RNA to pair, and then it'll make precise cuts to cut the genome. Both strands of the DNA will be cut, and the enzyme will, for the bacteria, destroy the virus. So the bacteria, what it's doing is it's taking a new viral infection. If it survives, it's now chopping the virus up into pieces and saving those pieces so that if it gets infected again, it's got a way of destroying the virus before the virus can get going. So it's what we call adaptive immunity. And it's exactly what your Norton antivirus software does. Okay, so if you don't update your computer's software from time to time, then a virus that infects somebody's computer might get into yours and destroy your whole computer. So you better update it, because the bacteria, just like us, are faced with the same thing. There's an application downloaded from the internet, right? Do you want to open that or not? Bacteria make that decision every day, and for them it's a life or death decision. So they use a system to, just like your antivirus software doesn't have the whole viral code, it has enough of the viral code so that your software can recognize that the application has this virus in it. So it's amazing how, we probably couldn't understand living things if we hadn't ourselves been dealing with information now for the first time in a way kind of similar to the way that living things have been dealing with information for literally billions of years. They are truly, truly sophisticated. All organisms are, and we still have an awful lot to learn about them. CRISPR is amazing because now, using CRISPR, again, we've figured out how to use this as a tool to modify organisms. It's allowing scientists to easily modify the genetic material in any organism, easily, cheaply, and it's all programmable. So it turns out if you express this enzyme in a human cell, humans don't normally have it, whereas the thing that Andy Fire and humans do have. So it was really easy because all we had to do is give a human cell a synthetic query, and the human cell carries out the search for us. Humans can't do this search because they don't have this enzyme, but if you give the human cell that enzyme, and a guide RNA that will take that enzyme to a particular genomic location inside of our DNA. This enzyme will precisely cut your DNA, and then you can do whatever you want. You can change the gene, you can put a new gene in, you can mutate the gene, you could repair a broken gene. There's a million different things you can do. And it's now taking the world by storm. We have the genomic sequence for humans. We have thousands of disease-related alleles identified in humans, and now we can actually fix them. And we can potentially use it to fix stem cells and repair a genetic defect in a human. It's really powerful. It's very potentially transformative for medicine. But even more importantly, now we can take human disease-related alleles and move them into other organisms. So you can take organisms that are not even traditionally thought of as model organisms, like C. elegans, can now become the recipients for genetic modifications that will allow us to model a human disease in that organism so that we can study, because you can't do a lot of things with humans that it would be unethical, but with animal models you can do some experiments that may lead to breakthroughs and will lead to breakthroughs for medicine. It's really, really huge. And another reason why you guys all need to know about gene expression is because we can modify the human germline using this tool. It's not safe yet, but that doesn't mean that people won't do it. It's easy to do now. So eventually, I expect to see genetically modified humans, not just animals and plants. So lots of things to think about. It's coming. All right, I'll be really quick because I'd like to have room for questions. I was going to go into, because I wasn't sure, since it's the day of biology, I didn't know how many biologists would be here, but I think most of you are not like hardcore biologists, so I'm going to kind of go quickly through our work, but just to try to illustrate one point that I think is really, really interesting. So when we cloned the first of those search engine proteins, it was this one, RDE1. And the way we found this gene is it's when we knocked it out. That's why model organisms are powerful. You can do things like mutagenize them. That's like treat them with chemicals that just randomly damage their DNA. And then you can find genes that are important for some function by screening the mutants for their phenotype, their defects in what they're able to do. So we found this gene RDE1 that fails to do RNA interference. So we would give them RNA, well, I never even told you what RNA interference is. It turns out just like viruses use double-stranded DNA sometimes, they sometimes use double-stranded RNA as their genome, because RNA and DNA can carry out very similar functions. So if you give a cell double-stranded RNA, it thinks it's getting infected, and it starts an antiviral response. And this gene, RDE1, is involved in that response. So when we knocked out this gene, the worms were totally fine. They didn't get sick or anything. But if you infect them with a virus, then the virus grows really, really well and makes them very sick. So it's an antiviral pathway gene. And why we were really excited about it is because now with genomes, we know the genomes, all the genes of every organism. So we looked at this gene, and we saw that the worm didn't just have one search engine gene, it had like 25 of them. So it had lots of different genes that were related to each other. And interestingly, here's a couple of them here. These are homologues of RDE1. We call them homologues because they're similar to this gene. And they're also similar to human genes. So humans have 8 members of the RDE1 family. I told you that we don't need to give humans this because they already have it. Humans have 4 on this part. This is a family tree, if you will. The closer you are on this tree, the more similar your sequence of amino acids is to each other. So these human genes, you can see how they cluster. The ancestor of humans duplicated this gene 4 times. The common ancestor of worms and humans had this gene also. And you can see there's still, these genes are very close to the human genes. They're further from RDE1. And if you go way over here on this other side, there's 4 more human genes. These are genes that are very important in the germline. And we're studying them now in the worm, but worms have members of this gene family too. So it's really interesting, right? You can look at this and you can see the common ancestor of worms and humans, which lived about 1 billion years ago, maybe 800 million years ago, really long ago. That animal already had this family of Argonauts and this family of Argonauts. Argonauts is just the word we use to describe them. It's just their name. So humans have them here and here. And then worms have a big bunch of them that there aren't really close human homologues for right here. This is a family of proteins that got duplicated in the genome. The piece of DNA was copied an extra time, and they ended up with more copies of this gene. And so we wanted to know, well, what do all these different search engines do? What are their functions? What searches do they carry out inside the cell? And interestingly, we found that many of them localize. These are 3 different members. They localize in these beautiful these red circles here that surround the nucleus of the cell. That's the DNA on the inside. There's this beautiful halo of these Argonaut proteins, the Caesar-1 protein, positioned right around the nuclear periphery. And so they're sitting right where they would need to be to see any mRNAs or transcripts that are coming out of the nucleus. So when the gene is expressed, the green blob copies it in here, then it has to get out into the cytoplasm, right? So it's got to go past this protein. Well, this protein also localizes in those same perinuclear structures. You're just seeing it here colored in green. And here's another one. This is the peewee Argonaut from over here. It has these human homologues, and it's also in these structures. And we just recently figured out what these things do. And I'm going to actually end just by telling you about them and not showing you any data, because I'd love to have time for questions if people want to ask questions. So it turns out that these guys encode a memory system. This Argonaut is actually a memory of all of the previously expressed genes that were expressed in the past, in the last generation. So I'm realizing I'll probably have to show you 1 or 2 more slides just to make that clear. This Argonaut is a memory of what we would call non-self. This is an Argonaut that is involved in silencing genes like viral genes or transposons, the sort of selfish DNA elements that hop around in the genome. This is required for silencing that. And this Argonaut is a scanning Argonaut. It uses small RNAs, and it allows misspelling, mismatches, so that it can scan thousands of different sequences. And it interacts with a genomically encoded small RNA called a peewee RNA. Humans have millions of peewee RNAs in their germline. Worms have about 30 or 40,000 different ones. But they're very sequence diverse, and they're constantly searching for foreign information. And this is what this really is an immune system that recognizes nucleic acids. And one of the key things, if you will, that an immune system must do is distinguish self and non-self. This memory of self is therefore essential for this process to work. So these RNAs are protecting the self RNA from the scanner, because otherwise the scanner would find and destroy the self messages. Once this finds something that's not protected, it can initiate a silencing reaction, and the silencing memory is loaded on this. And just as I showed you, there's a polymerase involved in replicating the DNA. There's a polymerase that copies these memories. It's called an RNA-dependent RNA polymerase. So once you form this memory, you can propagate it. And these Argonauts, they're in the germline, so they're transmitted in both the egg and the sperm. So the animal's not only sending its DNA from one generation to the next, it's sending an RNA memory of both active and silent gene expression in the egg and the sperm from one generation to the next. It's unbelievable. So if you're a bacterial cell, you wait until you get infected, and you create a memory of the infectious agent so that you'll be immune. The worm is actually trying to get a leg up on the viruses by remembering the self gene expression so that when they get infected, they can recognize the foreign sequence innately by using this scanning factor. And here, this just shows you that you can also introduce double-stranded RNA. This is how RNAi works. You can trigger the same kind of machinery using double-stranded RNA that gets then loaded onto this upstream argonaute RD1, and it recruits the same RNA-dependent RNA polymerase so that once it recognizes a target, it recruits this. This makes copies of the genetic information in the target that it wants to silence, and these form memories in these argonauts that I showed you in green, the silencing argonauts, to maintain and to propagate that silencing. So I think I'll stop there because I don't want to go too long. I had a bunch more slides that show you some of the data that supports this model for how we think the animal is propagating these RNA memories to essentially send information from one generation to the next. But I think the main take-home message is that, first of all, genetics, genomics, gene expression is really simple. It's really clear, it's very simple. Everybody should know it, everybody should understand it and feel comfortable thinking about it because it really is very simple. If you can read, you should be able to understand gene expression. It's very, very simple. So be encouraged, go out there and learn about that. But organisms can do amazing things with this relatively simple system. And there are what we call emergent properties. These proteins are so complicated, so interesting, and capable of so many different kinds of chemical and chemistries that there's really a lot for us still to learn. And the tools have never been better. Information now is flowing from the clinic into the laboratory. And in the laboratory, we can now model human disease faster and better than we ever could before. And they're real powerful tools now for, as I mentioned, with CRISPR, but also with other mechanisms to do, to do new types of therapy that are based on genetics. So it's a time right now where we really, I think, need to educate people about what's happening. As scientists, we don't educate the public enough about what's happening, and I think that creates a lot of fear, because when you don't understand something, you're naturally more fearful about it. But also there's a real opportunity to make new therapies, make differences in people's lives. And I think the public needs to be aware that if we do step up now and increase the funding support for basic science and for science in general, we can really start in the future to see big impacts in the laboratory and in the clinic. So with that, I think I'll stop. I'm looking forward to the day. It should be really fun and interesting, and I'm happy to take questions. There's a microphone here if anybody has a question. Thank you.
Hi, thank you for the presentation. I had a question about therapeutics. I was wondering if you might speculate what you think would be the most likely therapeutics that could be used, like with RNA interference, and is it actually being used today for that purpose?
Yes, that's a great question. It is. It's now in phase 3 clinical trials mainly for liver disorders because liver is easy to deliver the interfering RNA to. So it's taken a while for it to get to where it is, but it's that's another thing. It's hard for people to appreciate how long it takes for a discovery to get from the laboratory into the clinic. It took a really long time with monoclonal antibodies, and it's taking a similarly long time with RNA because we still don't understand the natural transfer of RNA and DNA from cell to cell very well. It happens, and viruses can help us do it, but we have a lot to learn about delivery still. So even though in the laboratory we can get very robust effects every time, in a human setting when you try to deliver, it only goes to certain tissues. And so a lot of the targets that people have had good success with are liver targets, and there's some therapies that are coming in the near future that will hopefully help patients in that area.
Great stuff. Thank you very much. So I live in both the clinic and the lab, and in my clinical world, we do pre-implantation genetic diagnoses. That took time after IVF and the like to be accepted. And that would seem to me to be where CRISPR is going to first be applied. But I'm wondering, given the dust-up over what's being done about mitochondrial disorders in the UK, are we going to learn from that experience? And then is that going to inform the first applications of CRISPR technology? I wonder what your thoughts about what's going on with mitochondrial diseases.
Yeah. I mean, gene therapy has had a very sort of bumpy road, and partly because people have gone into the clinic too soon and without really understanding all the biology well enough. And that's a real danger with CRISPR. It's still very, very early. The first papers on using CRISPR as a tool were published, I think, in 2012, and it's very, very widely adopted. There's several companies now that are trying to commercialize it for human applications. There are, I alluded to the possibility that there could be germline modified humans. There's already germline genome editing ready mice, basically. They're mice that are expressing this bacterial gene in their own germline, so that all you have to do is give them a guide and a donor. You can modify the mice to make them express any gene you want. There are going to be a lot of things like that. Should there be a genome editing ready human in the future? Maybe, I don't know. But it's still, there's a lot of off-target effects and unintended consequences that we have to sort out. So there's a lot to learn, and we should proceed very cautiously. One problem with genome editing any animal, but a human especially, is you can't get consent prior because they're not born yet. So if you're going to modify someone's stem cells, that's, I think, something that's a totally different story. But the germline, I don't think consenting parents is sufficient, because you're affecting the child, and that's anyway, please.
Hi, thanks for the presentation. I'm just wondering if you have any words of advice for the students in the audience who are interested in biology and research and want to kind of work on these hard problems. How do you get from being a student to being this kind of world-class researcher?
Well, what my advice is, follow your passion for, and don't be afraid. I think one thing students do is they tend to get the feeling that it's like all been tried and like it won't work type thing. You can psych yourself out. You shouldn't really believe anything you read. And if you find a good system that you think is interesting, like I got sucked into this worm, and I still work on this worm as my full-time job, believe it or not. But a lot of times people say to me, did you know what you were working on was going to be important before? And I was like, yeah, I knew, because they thought it's a worm, how important could that be? But the fact is that these mechanisms are so ancient and so highly conserved that you can learn a lot from these animals, these tiny microbes, even yeast, bacteria. They have a lot to teach us. CRISPR wouldn't exist if it weren't for bacteriologists who were doing very, very basic science. And so again, it just sort of makes me say again, pursue your interests, your passion, and follow it where it takes you. My mom and dad told me that. My mom's an artist, and actually I showed her those pea granules, the little germline. They're so beautiful. They're around the nucleus. And she's like, Craig, work on those. Those are going to be important. And that was like before we figured out the Argonauts were all in those structures. She had that artist instinct, something that beautiful has got to be important. And I think that's why, biologists and scientists they're actually very spiritual people, because to be a good scientist, you need to be driven by passion. So follow your passion. Don't try to be logical about it. Go out there, and if you think it's cool, work on it. and pursue it, and you're going to find something really, really neat. And you'll also be good at it because you love it.
That was I loved that last statement. That was super cool. So this memory thing is just awesome. Do you have any sense of how well conserved the function of this is across animals? Is this something that's happening everywhere, or is this something that might be specific?
Yeah, that's a great question. And you know, we get that all the time when you model organism, when they find something new. We get that question all the time. And now we're getting more and more confident about our answers to that. Because it turns out that usually these things, if a worm can do it, you know, humans admittedly are not as sophisticated as worms, but maybe they can do it too. But I think that the concept of keeping track of all the gene expression, using RNA as a tool for doing that, or maybe using other strategies, is something that cells will do because it's very powerful. As you can imagine, you know, and we're going to have a panel later on about aging, right? So when you are born, your genes are all newly programmed. Your tissue, you have the same genome in every cell, but it's going to carry out different functions. It has to remember which genes are supposed to be on in that particular lineage for your entire lifetime, right? And you live a lot longer than a worm. They only live like 2 weeks. And so, but they program their germline to keep this germline memory for billions of years. So it's really cool. So that we call it epigenetics, which is just another bit of, you know, meaningless terminology, but it's to distinguish it from the genetics. The genetics, the genomics, that's the hardwired stuff. That's what everybody has in every one of their cells. And then how you express it, that's different in every one of your cells. So when you differentiate your cells into different types of cells to make your neurons, your muscles, et cetera, you're programming them. And that programming has to last a really long time. So it's a really exciting field of biology where we're trying to figure out, you know, what are cells capable of remembering and how do they remember it? And it turns out RNAs play a big role in that. And all of these pathways, I didn't get to it in my talk, but the RNA memory pathway feeds into another memory pathway that has to do with the compacting of the gene into chromatin. And even like every organism where it's been looked at in several different kingdoms of life, we see the same sorts of proteins and RNAs collaborating with each other to reinforce each other in programming the DNA. So the DNA, in a way, you know, the amazing thing is the DNA is just sort of almost like the messenger carrying the information from one generation to the next. And the RNA is the thing that's sort of actively making it do what it needs to do at the right time. So it's really, you know, it's turned biology upside down. It's really neat. Okay.
Thank you.
Sure.
Hi, and thank you so much for the talk. As a biology student, you hear about Craig Mello from your day one, and so it's a great honor to hear you talk. So my question, I think it's a little bit too long now, but so one of the good and bad things about siRNA is that its effect is temporary. So it's bad because you know, it's not permanent, but it's good because if it does something you don't want to, at least it's not going to last for too long. And CRISPR does the complete opposite because it completely knocks out a gene. So how do you see that in terms of therapeutics? Because I feel like, would CRISPR be more risky in that sense, knocking out a complete gene? And then the other question that I have is, what exactly is the difference between, or why is CRISPR better than Crelox mechanisms, for example? How is it easier to be implemented in a human setting?
Yeah. So your first question is a good one about the benefits and the you know, the weaknesses of the two. They both have their strengths, as you pointed out, and you know, one of the nice things about the RNA interference is that you can have a therapeutic effect, and then when you withdraw the drug, you're not going to have it there anymore, and it'll go away over time. That's good in case you made a mistake, right? With CRISPR, you know, you're going to cut the genome, and if you cut it in the wrong place, you're stuck. So yeah, you're absolutely right about that. But yeah, there's lots of different ways. Those can be both good and bad, and there are lots of applications, and we have a lot still to learn about that. They're great. They're both really powerful in the laboratory as tools. I won't go into that a whole lot more because I think it's pretty obvious to people. And the last part of your question?
It's the difference or the advantages of CRISPR versus Crelox or other
Oh, yeah.
knockout
Well,
systems.
then the great thing about CRISPR is it's programmable, right? So you can artificially make a different RNA. Like, this is really cool. These RNAs are short, so you can synthesize them in the laboratory. You can make them very easily, and then you give them to the cell, and along with this enzyme, it'll then guide it to a new location. So instead of having to design this, every time to cut a particular region, you can do this very, very rapidly. So yeah, it's very, very easy to use. That's the main difference.
So let me just, before we close, thank you for a wonderful talk. Thank you all. And a little token of our gratitude. Thank you.